Hello and welcome back to Advancing Eco-Agriculture's series. This one is going to be a little bit different because this is going to be done a little offline. So I'm going to be making this presentation as well as the next one done a little bit differently. The first one, for those of you who are there, this is actually a four-part series. The first one has already been put up, and this is now the second one. As you can see, the title of this is going to be Toward a New Theory of Insect Olfaction. During the first presentation, we were just looking at the current theory of insect olfaction, and I spent some time discussing the merits and the demerits of that theory. And so now it is time for us to move on to a little bit of what we believe is going on. My name is Tom Dykstra, and in this series, I'm going to be talking about insect olfaction. I have been studying insect olfaction for many years now. This is something that has been a relatively big part of my career thus far, and I'm happy to be giving these series of four presentations, which will culminate in the fourth presentation, which I believe is going to be on August 30th. So I'm going to assume everyone, and I realize not everyone has, I'm going to assume that everyone has seen the first presentation. I don't plan on repeating a lot of it, so it's going to go quick. And for those of you who feel that you need to view the first one in order to make this a little bit clearer, please do so. But for those of you who have already seen it, I think it's time for us to begin. So toward a new theory of insect olfaction, we're obviously moving away from the current theory of insect olfaction that I discussed during the first presentation. Okay, so there we go. And there I am right there in my microscope. And I am AEA's scientific advisor. I have been that for just over a year right now. They've been tolerating me for that long. I'm with Dykstra Laboratories, been doing that since 1997. So it's been about a quarter of a century. And my contact information is down there. And we're now ready to get into the gist of the presentation itself. First of all, olfaction in insects, how is it accomplished? It's a question that I asked during the first presentation. I'm going to start answering it now a little bit more clear than the first time, because last time I was more or less attacking the current theory, and now I'm going to be setting up how insects are actually smelling. For those of you who are not aware from the first presentation, it was in 2016 that we believe that we deciphered the insect olfactory code, and these presentations are now helping us to understand how that deciphering occurs and how it all comes together, at least for the insect. So I had briefly mentioned that the sensilla on the insect antenna, there are sensilla, of course, on the maxillary polyps, labial polyps, and other parts of the body where chemoreception occurs, but these on the antenna are certainly the key players when it comes to olfaction or the smelling of odorants. And I mentioned during the last presentation that these sensilla, and this is a cross-section of the sensilla that you see off to the left, that there was a certain amount of time that the pheromone needed in order to reach the dendrite. It had to come from the outside, pass in through these tiny pores of the insect sensilla, then make its way through this watery matrix, make its way to the dendritic membrane. And I mentioned the 7 steps that are necessary in order for it to reach it, in order to activate the receptor. And during that presentation, I made it very clear that there was no way that this could happen, and that this wasn't just unlikely, it was actually impossible. So because it was impossible, the whole theory really has to be scrapped, because there was no way that I could get the odorant there in a timely manner. Now, I did know this probably about 2003 and 2004 when I became convinced that something else was going on. But as far as what that was, that came certainly usually after that date for the most part. All right, so the early conclusions that I drew is that the pheromone either binds with the receptor or it does not bind with the receptor. These are the only 2 choices that can happen with insect olfaction. If binding occurs, I had mentioned that technology has not found a way to show this yet. If binding does occur, detection of the pheromone must occur through some alternate means. What is that alternate means? I mean, obviously, it's going to have to be done without binding, and so now it's going to have to be detecting it from a distance, which complicates things, as you might imagine. So there are two main theories that are out there in regards to olfaction. One is the lock and key hypothesis of olfaction. And that is simply that it's a pheromone, for example, or any odorant locks itself into this blue receptor right there, and activation occurs with physical contact with the receptor. This lock and key hypothesis has been well delineated. It's well understood in regards to neurotransmitters and many other biological processes, but it has not been successfully applied to insect olfaction. The other theory is the vibrational theory of odor. Many of you have probably heard of the vibrational theory of odor. I will not be talking about electron tunneling. I will not be talking about the card swipe model, but I'm going to be talking about essentially a theory of odor that is akin to the vibrational theory of odor, and we'll just call it that for simplicity. And that is, if you have an odorant as designated by this white oval right now, in that an insect scintilla, represented by this triangle, would be able to detect it from a distance. And if it was going to do it from a distance, it would have to be composed of some electromagnetic frequency, because there's really no clean way to do this without electromagnetics, since there is no contact. So this is activation without physical contact. We know that activation occurs. We know that the odorants are going to activate the receptors. This is, I think, well established. But how this occurs is not that clear. And now I'm going to introduce you to a new theory, which is based upon the vibrational theory of odor. And so it's a good idea, and in order to acknowledge those who went before you, the vibrational theory of odor. Is this something that Tom Dykstra came up with? Absolutely not. I think a lot of you know this already. GM Dyson was really the first one to put it in paper, and that was back in the 20s and 30s, and he had just a few publications. Didn't go into a lot of depth out there, but he was really the first one to talk about this, and then it kind of died down for a few decades. Robert Wright, who was based up in Canada, really took off and did this, started in the 1950s and worked through the 1980s, published a lot. It is to him that I attribute deterrent frequencies. He was the one who introduced that term. I will not be talking about deterrent frequencies during this presentation, but he really set up a fantastic foundation that we could test and understand how insects were smelling a little bit better. And then Philip Callahan came a little bit later. They were contemporaries with one another, but Phil Callahan was in Florida, Robert Wright was in Canada, and so there was not a whole lot of overlap, but occasionally they would have run into one another. And Phil published in the 1960s and the 90s, and he was taking a look at it from a slightly different aspect as well. But these were the three individuals with which the whole theory kind of began and continues to do so. And my job was simply to build upon the foundation made by these three gentlemen. So this is, it should be similar for those of you who've seen the first presentation. This is a cross section of the scintilla, and I've got pheromone, which is designated by these blue balls on the outside of the insect, and the cuticle is designated by these the really thick black lines. I have some pores in them because we do know that these pores exist. There are some tiny pores that exist that are, I think, somewhere between 6 nanometers to 65 nanometers. So they're very tiny pores, but they're there. And then we have the syncytial lymph, which is a watery matrix. And then we have the receptors. Now, as I mentioned, activation occurs. We know that the pheromone can be detected by these receptors as designated by a dimer receptor off to the left of the screen. So if we know that activation occurs, and yet I've shown that physical contact is not occurring, we've got an issue. We've got several issues, as a matter of fact, and that is, how is the pheromone detected if it actually cannot make physical contact? And there are 3 different issues that need to be addressed and will be addressed very briefly during this presentation. The first one is the interface of the cuticle. So the pheromone, if it's not getting inside the actual scintilla itself, is sending a message. We believe it's a vibrational message, and it must make it through the cuticle in some fashion or another. It must collect that energy, and it must pass it through. That's the first thing that we need to address. The second part is the syncytial lymph. The syncytial lymph, it's a rather large area, watery matrix. It's in between the pheromone on the outside and the dimer receptor on the dendritic membrane on the inside, and we have to get through that as well. So that needs to be addressed as well. And then finally, we need to get to the receptor itself, and that is how is the receptor actually activated, assuming that the message gets through the first, which is the cuticle, gets through the second, which is the syncytial lymph, finally makes its way to the receptor. How would it actually activate it? And so I'll very briefly be discussing those. We'll discuss them in more depth in presentation number 3. So the first one, what do we know? Erickson in 1975 found that the mean surface potential of honeybees varied throughout the daily flight period. What he found is that bees that were leaving the hive had a charge on the outside of their body between negative 14 millivolts to plus 8 millivolts, which if you average is running right around 0. So essentially this means that they're not charged when they are inside the hive. But the returning bees are. And they have anywhere between plus 13 millivolts to plus 59 millivolts. So the charge difference is huge. And you can see that there are no negative charges when the bees come back into the hive. It's all positive, and it's relatively high too, at least compared to the negative 14 and the plus 8. So what is happening right now is that the bees are being charged through flight with the beating of the wings, with the friction of the air, and this charging of the body is something which is happening with the bees. We know through biophysics that this is going to be happening with all insects, but we're just showing this as far as honeybees are concerned, is that so long as they are moving and flying, and this is how most insects smell, most of them are smelling in flight, is that they are charging the outside of their body and that there is a positive charge associated with it. This is, let's see, what we're showing here. This was a German researcher, Ulrich Wernke, who was taking a look also at honeybees as well. And he found that abnormal wing fluttering occurs in a group of bees in a Faraday cage. A Faraday cage means they were excluding many of the external electromagnetic frequencies. And the fluttering is associated with autoelectrification. How do we know this? So you can see the large lines on the left side of the screen, and this is the abnormal wing fluttering that is occurring. As soon as the cage is supplied with atmospheric ions, which is that large arrow at the top, that abnormal wing fluttering ceases instantaneously. So they can go back and forth, and they can play this game again and remove it, and the wing fluttering begins again, and then they can introduce the ions and the wing fluttering will stop. And so this autoelectrification effect can actually occur inside the hive as well, and not just when it's in flight. But you can see the importance of this process and the importance of ions, especially on the outside of insects in general. We also know from 1984 that the fixed negative charges are going to be present on the inner cuticle of the syncytiae. The inner cuticle is different from the outer cuticle. I was just talking about the outer cuticle, and I was talking about these positive charges. But on the inner cuticle, Kyle found that there were negative charges, fixed negative charges present on the inner cuticle. Well, this is interesting because if we have fixed negative charges on the inside of the cuticle and we have positive charges on the outside, we have now created a differential, an electrical potential, a differential between the outside and the inside, positive on the outside, negative on the inside. And this is important because we have now created what is known as a dielectric. A dielectric just means two electrical charges, and that's where the die comes from. So these two electrical charges are opposing one another. They're positive versus negative. And these can be maintained as long as you have a dielectric substance in between them, which in this case is going to be the cuticle, and it actually is the wax as well. The wax is actually helping out because it has electret properties. So because we know this is happening right now, we now have a dielectric situation. And so because of this, it's good to take a look to see what do I mean by a dielectric. And it's directly opposed to a conductive material. Many conductive materials are made of metal, and many antennas are also made of metal. Everyone, I think, is familiar with the metal antennas that people put up on their houses, or at least they used to. I'm not sure that's done so much anymore, but I know there are a lot of ham radio operators. I know a few myself, and they're still using metal antennas, but it's a conductive material where positive and negative charges are allowed to pass through it because it's conductive. This is different than a dielectric material. A dielectric material does not allow charges to pass between them, which means they can maintain a charge differential on opposite sides of the dielectric material. So here you can see that there's a largely positive charge on one side of this imaginary dielectric material and largely negative charges on the opposite side of the dielectric material. Well, this has repercussions for us because dielectric and conductive are two different forms of antennae. And one dielectric that I think everyone is familiar with is polystyrene. Polystyrene has been known to be a very good dielectric, and obviously it protects your hand from the hot coffee if that's what it's for. But they also knew about dielectric antennas back in the 40s, and they often use, and at times they were using polystyrene as a type of dielectric antenna. So I just want you all to be aware is that there is a little bit more out there besides just a metal antenna, and a dielectric antenna has been known since the 40s. So it's been around for a long time, and they are useful. They have certain properties associated with them, and it's these that I need to focus ever so briefly on during this presentation. So this is a good dielectric antenna. After you drink the coffee, you can turn it upside down and it will actually be a better antenna because it should be sloped towards the top, especially if it can come to a point, it will form an even better antenna. But this cup could be used as a dielectric antenna, even, although a crude one. So the properties of a dielectric antenna, there's only two. As long as these two conditions are met, you can have a potential dielectric antenna. One, it needs to be composed of a dielectric material. So that's one of those dumb moments. But two, the dielectric material must have an effective dielectric constant. Well, what's an effective dielectric constant? It shouldn't be too high and it shouldn't be too low. So the waxy layer on the outside of the insect has a dielectric constant between 2.5 and 3. We've known this since at least 1967, probably earlier. This just happens to be when it was first reported. And it's similar to the dielectric constant of polystyrene, which is 2.55, which is an excellent dielectric. And as I mentioned, can be used for picking up electromagnetic frequencies. So these two conditions are the only things that need to be met in order to have an existing dielectric antenna. However, it does get more complicated because it's not just about having, an upside down coffee cup. So you can have a dielectric rod antenna, which would be off to the left, but you can make it better. And you can make it better by tapering it to a point, as I alluded to a couple slides ago. You can also make it even better if you take the diameter of this dielectric rod, which is now tapered, and make it thinner at the bottom. That will actually make it a better, more efficient antenna. And you can also lengthen this same antenna, and this will actually make it even better. What this does in effect is it increases the directivity or gain of an antenna. And this has been known as reported, since 1952. So all of this is old information. Some of you may be familiar with it. Some of you, this may be the first time that you've heard of something like this before. And so if we go back to the insect, we see we've got a close-up, a scanning electron micrograph of an insect antenna. There are a number of dielectric antenna, but I was sensilla on the outside of this, posing as dielectric antenna. And so you can see that they are relatively thicker at the base, and they get thinner as you get near the tip. We also can see, it even happens with the Coeliconica. The ones that look like little circles down there, they are also pointed at the tip as well. We have a cross section of the sensilla off to the right, and you can see that there are two things, and I do want to point out those two important things, and that is the cuticle and the wax layer. The cuticle of the insect is relatively thick. However, When it comes to the scintillae, they are not that thick. As a matter of fact, they're quite thin. In addition to being thin, they're perfectly, they have a diameter which is almost perfect all the way up. So the cuticle on an insect varies a little bit based upon different parts of the body, but the cuticle on the insect scintilla does not vary that much. And as you can see, it is very thin. We still got it with the pores that I mentioned before. We stuck those in, so you can be clear that we're talking about the cuticle. And then we have the wax layer too. The wax layer is also generally a little on the thick side when it's covering the insect, largely for water loss. But this wax layer extends over the scintilla, and you will also notice that it's very thin. Just like the cuticle is thin, the wax layer becomes very thin. And in addition to being thin, it's of a uniform thickness all the way up the scintilla. And that is what I wanted to point out to you here. So properties of a dielectric antenna, we need only two, but I'd kind of like to know, what do I need for a good one? Because certainly there have to be some other properties. A third property, they need to be long and thin, or tapered, or rounded at the tip. And sometimes we do find these roundings going on. We see them in the Lepidoptera antenna, the clubbed antennae, and some of others. So this is the property as known by the dielectric rods, and is associated with the dielectric antenna. Four, in order to have a good dielectric antenna, it needs to be covered by a good thermoelectret, such as wax, which it is. A fifth characteristic is the directivity. This is for you antenna people out there. And I know some of you are going to be listening to this because I've talked to some already. The directivity is proportional to the tube length, characteristic of a dielectric rod, something we've known since the 40s. And so this is another characteristic which is also satisfied by the insect scintilla. If the wall is thin, it can achieve a single lobe radiation pattern, which makes it better, gives it more directivity, more gain, essentially making it a better antenna, which means it can localize where a message is coming from. And seven, the antenna and the thermoelectric need to be of a fixed consistency. If they are not of fixed consistency, the antenna will not work well. And yet we do have that with all of the insect scintilla. So not only do we have a dielectric antenna, but we satisfied 5 additional requirements for a good dielectric antenna, which means we're getting very, very close to showing the superiority of this system on the insect. But can we do better? How about a perfect one? Is it possible to make a perfect antenna? Some would say no. I would probably agree with them, but it has been reported that you can make a perfect antenna, a lossless antenna. And this can be done with a dielectric material, a dielectric antenna. In order to make a perfect antenna, many researchers have found and published that you need to make tiny perforations in order to create a lossless antenna. So if they started poking holes in a dielectric antenna, they could make it actually lossless. And this has to do with mathematics and physics. And it actually makes the thickness of the cuticle mathematically or physically zero, when in actuality it's not. So it's an interesting phenomenon. And yet, even though it is interesting, we also have that too. The insect scintilla, not all of them, but many of the insect scintilla have tiny perforations or pores. As I mentioned, they're usually running on the small side, 6 nanometers. They can go all the way up to 65 nanometers, but it's still tiny. And these tiny perforations exist on a lot of the antenna that are out there. So not only have we satisfied the conditions of a dielectric antenna and a good one, but we've even satisfied it as a perfect one. We're now faced with the reality that the insect has dielectric antenna, that they exist in they're very good, and that they might even be perfect. That is the truth that we're faced with right now. This doesn't say that the insect is actually using it as a dielectric antenna. It's just saying that all the conditions have been met for a dielectric antenna. And so that's all I needed to discuss right now in regards to that. I just wanted to show you, I tried to draw this to scale to the best of my ability, just to give you an idea how thick the pores are. And the pores cover between 1 to 1.5, 2% at most of the surface of an insect cincilla. So they are scattered. So most of the cincilla is composed of just cuticle. And there are some holes. These pores, very tiny, but they're spread out, and you can see that they're not that dense. It just gives everyone who's listening to the presentation a little bit better idea of how many pores there are in a particular cincilla. This one happens to be a Cincilla basiconica from Bombyx mori, which is the silk moth. And for those of you who are interested, you can go ahead and, because I'm going to skip this very quickly, but I just wanted you to see that the idea of this being a dielectric antenna was started by Grant back in 1948, the first one to talk about this. And it ends with 2002 papers where they were talking about perforating dielectric resonator antennas. And so if you wanted to take a look at, if you wanted to pursue this a little bit further, you can take a look at some of these sources, and you'll be able to read a little bit more about this. Okay. All right, so I've talked about the existence of this being a dielectric antenna. So we've now got a way to collect energy, which is a great start, but it's not everything. We've still got, 2 and 3 to get through. So I've got the cincillar lymph, and this at first glance seems to be a problem because we are told that this is a watery matrix. And I can tell you right now, I mean, if you want to transmit energy through, especially infrared energy through water, you're going to be in trouble because it's not very good. It's not a very conductive substance. It doesn't transmit all frequencies very well. Some frequencies it does transmit, others it doesn't do well. And so water is not really going to cut it as far as a good material to transmit any potential frequencies. But do we have that? Is there anybody who thinks that we have pure water in the cincillar lymph? I don't think we do, because we understand that this is a live animal, and animals are not dealing with, we just don't have distilled water in different parts of our body. The cincillar lymph is not pure distilled water. It's complex. It is a salt-based structure. There are lots of salts that are based in it, and these salts are important. Why? Because as soon as you introduce salt into water, for example, even if it's pure water, distilled water, what changes? The conductivity changes. The conductivity changes drastically. You can increase the conductivity, 2, 3, 4 orders of magnitude over what it was before if it was just pure water. And the cincillar lymph is salt-based. So we've now got an interesting situation here where we've now got the potential for a conductive fluid. So this is based upon many of the regular salts, predominantly sodium and potassium, but there's also some other potential salts that are in there, and they have charges associated with them. For those of you who are familiar with chemistry, you understand the ionic bonds are separated, and NaCl, or table salt, is split up into Na, sodium, and then Cl, which is a chloride atom. So the sodium has a +1 charge, the chloride atom has a -1 charge, and they break apart in water. And so you've now got these active charges moving around this watery matrix. It's a salt-based matrix. And so because of this, we now have salt, which means we now have a conductive substance, and we're now getting a little bit closer to understanding how insects might be smelling now. So I've also mentioned to you in the last presentation that the pheromone binding proteins are charged as well, and that most of them are negatively charged. We've run through, I think, over 150 of them, and about 81% of them were negatively charged. Some of them were positively charged. We did have a few that were neutral, but you can see that there are some additional charges inside the cincillar lymph. It's just not about the salt, is that these proteins seem to be contributing charge to the matrix, and this does seem to be important. So I want to mention it right now because I'm going to talk about this a little bit more in the presentation. So here's our situation right now. We've got this medium right now, and this medium is composed of a high concentration of these binding proteins. We've got a salt matrix in the cincillar lymph. Then we have these binding proteins, which are also charged too. And so we've now got a potential situation right now where we might be able to start transmitting electromagnetic frequencies across the cincillar lymph. But is that the whole story? do we know for sure that this salt solution is important? And I would contend that we do. So if you take a look at decapod crustaceans, and this would include lobsters, it includes the blue crabs. Deca means 10, these just 10-legged crustaceans. They're found in a marine environment. I think we all know that. So they're in the ocean. And you can take a lobster and remove it from its saltwater environment and place it into fresh water. And you can essentially flush its environment with fresh water, and it will cause them to become anosmic for about 1 hour, maybe 45 minutes. So during this time, you've essentially cleaned out the decapod crustacean's sensory abilities for its ability to smell for about 45 minutes to an hour simply by flushing it with fresh water. This is interesting to know, and then it's restored. And after about 45 minutes to an hour, it's restored. And then usually further studies can be done. Many researchers will then do further studies on these decapod crustaceans, especially spiny lobster. And so that is a fact. We also know, some of you may be saying, well, what about Calanectes? And I would say, yes, what about Calanectes? Good point. So Calanectes is the blue crab. It is found in a marine environment, but it also migrates up the Chesapeake Bay and other areas. It migrates up into a freshwater environment in order to breed. So what's the story with this? Because now we've got a crab, the blue crab, a decapod crustacean, and it's in a saltwater environment, and then goes to a freshwater environment, and then it goes back to a salt. So what's the deal with that? And that's an excellent question. So blue crabs survive in both freshwater and marine environments. They need to, because as I mentioned, they will often, the mass migrations at the Chesapeake Bay, at the base of it there's a lot of fresh water there. And they have the ability to move salt solutions into and out of their syncyllae, depending on their environment. So they're able to create an ionic microenvironment with their estatasks, which are the olfactory receptors. They're kind of akin to the olfactory receptors that we find on insects. So this salt solution is moved up into the olfactory receptors as they move into a freshwater environment. And then as they go back to the ocean, the salt is removed. So this brings up some good questions, and that is, what is the deal with these pores? Arthropods, which include decapod crustaceans and insects, what's the deal with their syncyllae, and what's the deal with their pores? Well, what do we know about them? Well, terrestrial insects, as I've kind of alluded to during this presentation, terrestrial insects, do they have pores or do they not have pores in their syncyllae? And for the most part, they do. They do have pores in their olfactory syncyllae, as you would expect them to. Now, what about the crustaceans in a marine environment, so a salt-based environment? What about them? Do they have pores in their olfactory syncyllae? Yes, they do. And the reason why they do is because the marine environment, a salt-based solution, is going to slip through the pores, and you're going to be able to maintain a salt-based solution inside their sensory structures. So they don't really need to exclude it. It's not really an issue. What about aquatic insects? Aquatic insects, as I think most of us are aware, are exclusively found in freshwater environments. They are not found in the ocean. You do not find them in, generally speaking, marine environments. There are a few exceptions, but they're not important to mention now. And these aquatic freshwater insects, what's the deal with their syncyllae? They have no pores in their olfactory syncyllae. And this should make sense as you're listening to me right now, because if they did, fresh water would be able to get in, and it would make them probably anosmic, which means they wouldn't be able to smell again. And so they need to exclude fresh water from getting inside. They need to maintain, obviously, a salt environment inside it, and then they need to exclude the fresh water so that they can still smell while they're under the water. And these aquatic insects include mayflies, dragonflies, stoneflies, insects of that sort. But what about these same aquatic insects when they become adults? Because they leave the water and now they're flying around. Mayflies fly around as adults. Stoneflies fly around as adults. Dragonflies, damselflies, they're flying around adults. What happens to their sensillae as terrestrial adults? They regain the pores again, as you would expect them to. So once again, they have the pores just as the other terrestrial insects do, but the same insect in its aquatic stage didn't have them. And the same species, once it comes out of the water, now has pores in its olfactory sensillae. So it's certainly not a species thing, because they can do that within species. Well, this is really interesting because we've now got some evidence right now that salt-based solutions are important for smelling. And so this applies not just to insects, but it even applies to other arthropods as well, as I'm kind of alluding to as I'm bringing in some further evidence. So we've now got the second situation kind of covered right now. I'm going to be covering a little bit more in just a second and a few more slides, but I wanted to focus a little bit more on these receptors, the Dimer receptors, or these membrane-bound receptors, because eventually the message has to get to them and they have to respond in some fashion or another. So what's the deal with them? Well, they are membrane bound. Generally speaking, most of the olfactory receptors and the gustatory receptors are 7 transmembrane receptors, which means they happen to pass through the membrane 7 times. And a lot of these receptors have that 7 transmembrane characteristic to them. There are some 5s and 6s as well. But there are some other proteins which may only go through the membrane twice. And these are in invertebrates. These are the CD36 proteins, which only go through twice. They have a correlate in insects, and they are associated with the olfactory receptors. These are the SNMPs. The SNMPs are the sensory neuron membrane proteins, and they are similar to the CD36 proteins in vertebrates, where they only pass through the membrane twice. And so, but even though whether they pass through 7 times or whether they pass through 2 times, they're still bound in the receptor itself. So anytime they would be absorbing energy, they're going to be doing so in the context of being in a membrane. And so any energy transfer too is usually going to be, if it's close by, to some other receptor. So I just need you to be aware. And there are a lot of these receptors. Just on E. coli, one of the smallest cells that's out there, you're going to have maybe 60,000 membrane proteins. And that is a very, very small cell. So you can imagine that a much larger cell is going to have much more than 60,000 membrane proteins. So the take-home message is there's a lot of them. There's a lot of them. So in order to test these receptors, it's difficult to test them when they're inside the sensilla. And so we have been able to decipher them and determine what their amino acid structure is like, and we can reproduce it, and then we can insert these receptors into artificial organisms. So one of the common ones are the Xenopus Oocytes, which essentially are frog eggs. So the frog eggs, which are down there at the bottom, we can actually insert the code for these receptors, and they will express themselves in the Xenopus Oocytes, and we can actually do some olfactory tests on these eggs. Another option that some researchers are using are the human embryonic kidney cells from a human embryo, and they are then reproduced, and they are then also, you will also insert the genetic code into these human embryonic kidney cells, and they will then express them, and they can also do testing on them as well. So these are the main 2 which are used by predominantly most of the researchers looking at insect olfaction when they want to decipher what's going on with a single protein. But there is a third. It's not as well known and it's not used as much. And there are some reasons for it too, is because it's a different system than using a live cell. And these are the liposomes. The liposomes are essentially, I mean, it looks like a cell to a certain extent. It's just empty. It's empty on the inside. And it's composed of a phospholipid. It's a bilayer, just like you would find in a normal cell, but that's it. It's just a phospholipid bilayer. And so because of that, there's not much to it. It's just like an empty cell membrane And there's nothing inside. There's no nuclear membrane. There's no endoplasmic reticulum. There's none of that stuff. It's just empty. So it's just a ring of these lipids, which are, as I zoom in right now, you can see these are the circles with two extensions coming off that look like spaghetti strands, and they're opposed to one another. And that's why they form a bilayer, because there's actually two layers as they face one another. And these are liposomes that can be used in scientific research and have been used in scientific research to look at many questions in science, such as olfaction. Sometimes they can be big, like they are to the left, but you can also get some small liposomes with maybe 100 lipids, and they can form some pretty tight liposomes. But generally speaking, the size of it, it does make a difference, especially if you're looking at the membrane permeability. But I just wanted everyone to be aware is that you can get certain different sizes associated with them. And you can then do the same thing that we did with the frog oocytes or with the human embryonic kidney cells. And you can actually take, let's say, an insect OR, an insect odorant receptor, and you can insert it. And Japanese researchers, Nomura and Kurihara, did this back in 1987, so this is not new stuff. Liposomes as a model for olfactory cells. They looked at the changes in the membrane potential in response to various odorants. And what did they do? Well, you simply just take the protein and you can insert them, embed them into the liposome itself, and then start testing odorants against it to see how they react. And this would also give you an idea as to what the odorant receptors are responding to. So that's just some of the basic stuff that has been done. But it goes beyond that. You can take a random protein. It doesn't even have to be odorant related. It doesn't have to be an odorant receptor. It could just be any random protein. And you can insert that in a liposome too. And so this random protein, picture protein, can be inserted into a liposome, and you can actually get the liposome to respond to various odorants with this random protein. This is really interesting, but it doesn't stop there, because you can go on to even a further step. So Kurihara's lab, 1990, we're also taking a look at this liposome phenomena. And so, as I alluded to in the slide just before this, you can take a random protein and put it into a liposome and start to get reactions, and all that is fine and good. But there's a little bit more to that than that. And that is, if you have these proteins, you can introduce them to a liposome, and then you can also introduce other proteins. You're not limited by putting one protein into a liposome in order to start studying it. And so what they found is that you can add many, maybe even random proteins into a liposome, and they were actually able to detect, they had about a dozen odorants that they were able to successfully detect and differentiate with their ridiculous looking random protein liposome molecule, which is an amazing thing in and of itself. But there's still more to it than that. Nomura and Kurihara, 1989, 2 years after the 87 paper that I just mentioned, and before this one, looked at ions. So I was talking about the salt ions before. They looked at the similarity of ion dependence of odorant responses between the lipid bilayer and the olfactory system. Well, what did they find? They found out that if they used distilled water, just regular water, that they got no reaction at all, none whatsoever. But if they added sodium and potassium to the outside, not to the inside, the inside would still be can still be protected, but to the outside, they're actually going to get a blip. So they now expose an odorant to it, and you will actually get membrane permeability, to a detectable response from the liposome once you add salt to the outside of the liposome. Well, that's all fine and good, but what if you just go crazy and you increase the salt even more? What if you increase the sodium, you increase the potassium, so you've got a higher concentration? What happens then? The response goes up. And so the response goes up based upon the concentration of salts. This is very, very interesting stuff. Obviously has relevance to what I'm talking about. But now we've got the situation over here And they found that you can go one step further, and you can actually use divalent cations rather than using the monovalent cations, which have a +1 charge. We can now move to the divalent cations, which have a +2 charge. And they found out that these are very good at creating that salt environment, and that you will get a heightened response. And this heightened response is going to be even more so than it was before, but you can actually reduce the number of divalent cations and still get the same response to a monovalent cation. This is suggesting, I think you're all understanding this right now, is that the more charges you introduce to the system through these various ions, the better the liposome is able to play this game. Now, this is a game that we're not able to do with the frog cells. We're not able to do this with the HEK 293 cells because they're live cells, and you can kill them. If you put them into distilled water, they'll explode. With the liposomes, you can play these games, and we can take a look to see what these reactions are like. So this is all interesting because we've now got another system going on here that is largely salt-based. And so I just want to review this real quick as we kind of come to the last few slides. And that is, if we have a vibrational wave coming from a pheromone molecule, and it is going to get around an antenna, any antenna is going to do this. It is going to, usually the lowest impedance is going to be at the tip. And so that's where the beam will probably get picked off. And then it kind of wraps its way around, especially if it's a dielectric waveguide antenna. It will guide the wave around to the base of it. So an incoming wave will resonate to, be absorbed by, stick to. These are all different terms used in antenna technology to describe how the wave is picked up by the antenna. And here we have a sensillum and a Florida scarab mite. And you can see some of the ridges. So we talked about the dielectric wave guide because it will guide a wave on the outside of it. And oftentimes you will find these ridges on the outside of these insect sensilla. This is incredibly small. You can't see it. This is a scanning electron micrograph. So this is not visible to the naked eye. But a lot of these ridges are found on many of the insect sensilla. So if you're basing how detection occurs on the current paradigm, which is the last presentation that I gave, you would say that a pheromone molecule would hit a certain point on the sensilla, and it would only be at that point unless it was able to innervate on another part of the antenna and get through another pore. So that's how the theory goes with that side. But if it's based on antenna theory, which I'm asking you to consider right now, you're going to get a very different system. And the system is the whole antenna essentially is going to light up. It's not a point effect. With an individual odorant molecule, it's very much about the point effect because the point has to be mediated by an odorant, which will make it to a particular point on the dendritic membrane. Not so with an antenna. Antenna just needs to be able to be in close proximity, and the whole antenna, from a certain standpoint, lights up, which means it is now detected, assuming that there's a sufficient signal in order to be detected by the antenna. So therefore, if an insect is swimming through the air, which is in a sense almost what they're doing, they're flying through the air and they've got some odorants that they're picking up. These odorants only need to come into close contact with the sensilla, and this whole region of a pheromone, there can be thousands to millions of them, are going to be picked up by multiple sensilla at the same time, and they're all going to light up and they're going to tell the insect that I found, let's say, a pheromone, and if it's a male Saturniid moth, he's going to get excited and he's going to start flying off looking for the female. So this system is fundamentally different. As you can see, it's also, I think you understand, a much faster system than what I had discussed during the first presentation. And so it's an interesting system that is also based upon the salt matrix too, because remember, not only do we have the salt in the middle there, but these PBPs, or pheromone binding proteins, are found at about 5 to 10 millimolar, which, if I'm going to translate for non-scientists out there, that is really high concentration. So there's a lot of binding proteins in there. This is not just about sodium and chloride items adding to the conductivity. We now have pheromone binding proteins, and they're really strongly charged. They're about 40 to 70 charges on an individual pheromone binding protein, which is remarkable because it's a high concentration of charges, much more than you would expect through random chance, because there are only 5 charged amino acids, and yet these are 120 to 140 amino acids long, and they're running between 40 to 70 charges. So you can see that they have a preponderance of charges, and so we're simply adding to the possible charges. And as I alluded to, this does seem to be important as far as detection has occurred, in order to get as much positive and negative charges in there, make it a conducting fluid, and allow olfaction to occur the way that it was intended. So if we have the pheromone, as I've alluded to, on the outside, how does it actually get in? As I mentioned, there is a very good possibility of a conducting fluid. It will then reach a receptor. The receptor will pick it up, and actually, if there's enough energy, it will actually activate the receptor. And so this is an interesting system because this is being done from a distance. And because it's being done from a distance, as I've suggested, electromagnetically, this is going to change how insect olfaction is looked at, assuming that this theory is correct. And certainly the last one doesn't hold enough water. So I'm asking you to consider this theory, as especially as I go into the next 2 presentations, is for you to understand how something like this can occur. So if I have, and I've just done some random frequencies up here, if I have some random frequencies, and this is just, let's say, the frequencies coming from a given pheromone molecule, do they line up, for example, with the absorption frequencies of the receptor molecule? So if I have an imaginary receptor molecule, and these are the absorption frequencies, do they line up with one another? No, not a single one. So even though this looks pretty good, they do not line up. There's not a single frequency here which lines up with the top. Therefore, this situation on this slide is showing non-resonance. There is no resonant effect between the two. And as I said, we're kind of needing a resonant effect between the pheromone and the receptor in order for reception to occur. So if I have another receptor and I'm able to determine what the absorption frequencies are, do these line up? And in fact, a lot of them do. So in this particular case, in this imaginary scenario, we've actually got a lot of frequencies that line up before. And so now we have active resonance. It's not 100%, never is, but you can easily get 3 to 6 frequencies. If you're having a good resonant effect, you can have at least 3 to 6. Sometimes it gets to be more. I just did more on this one for effect. But you can see as that once resonance occurs, that we've now got evidence for an electromagnetic theory of olfaction. And so that is what I want you all to consider during this presentation, because this is going to set you up for what I'm going to be talking about during the 3rd presentation. And so I have looked at way too many. I mean, we're well over 10,000 spectrums over the past 6 years, since 2016. I've got spectrums that I have to look at in regards to pheromone molecules. I've got spectrums down on the right side, which are taking a look at absorption frequencies. And I've got to line these up. And I've spent a lot of time. And at first, I was a little flexible. Sometimes I would say, well, it's close. And I would count it. And it's gotten to the point now in 2022 where if it's close, it doesn't make the grade anymore. I now want something to line up perfectly if I'm going to count it as a true resonant effect, and then I can then determine what the insect is smelling. So hopefully this presentation has allowed you in order to see and understand a little bit better how an insect would be able to smell electromagnetic frequencies, which I can tell you is what it is doing right now. During the 3rd presentation, what I want to do is I want to introduce you to these resonances. I'm actually going to point out specific examples. And so you will be able to see, for example, just to talk to you about carbon dioxide reception in mosquitoes. We know that there are some carbon dioxide receptors, and we know what the carbon dioxide frequencies are, and do they line up? If they don't, then we don't have resonance. But if they do, then we do have resonance, and we're starting to determine how, for example, mosquitoes pick up carbon dioxide, or really any odorant molecule as well. So I think that does it for me. This is the final slide. I just wanted to give you a quick intro as to where we're going to be going for the third presentation. And then the fourth presentation will be almost exclusively on aphids. I'm going to be really zeroing in on how aphids are utilizing all of what I am introducing. And I needed everyone to have these three presentations so that you can understand where I'm going with the aphids, which will be the subject of the fourth presentation. So I think that does it for me. I would like to thank you all for paying attention throughout this entire presentation. For those of you who got this far, I hope that it was enlightening for you. I'm really looking forward, as I'm looking at this right now, towards presenting the third presentation, and that'll be in a couple of weeks. And until then, I think it's time for me to sign off because I've got a presentation to finish up. Thank you very much. Have a good day and enjoy the rest of your week. Bye-bye now.